Hi everyone, it's Kevin back with video number 2 in lecture 28. It says 23, it should say 28. Talking about photosynthesis. It's all pretty simple because we've talked about the principles before. We've talked about capturing electrons and delivering them to an electron transport chain. That happens in photosynthesis. The electron transport chain pumping protons, we've talked about, that happens in photosynthesis. The chemiosmotic gradient from the pumping of protons being used to make ATP, we've talked about that. That happens in photosynthesis. So a lot of the principles we've talked about. We add to that light from sunlight, exciting electrons, and we pretty much have it. So let's get going. This is what we were working through. You can stop and look at this. But we're going to talk about the light reactions of photosynthesis today in some more detail. Here they are in summary. Light comes in, excites electrons. Electrons are removed from water. So how does that happen? It happens by means of reduction potentials, and we will talk about how that happens. So electrons are removed from water. Water's oxidized to oxygen. So pretty straightforward. Those electrons are dumped on NADP plus to make NADPH. As a result of the movement of electrons, protons are pumped. Those flow on the right here back through ATP synthase, just like we saw in oxidative phosphorylation. And ADP plus PI, inorganic phosphate, makes ATP, adenosine triphosphate. So there's the big picture. Here's some more detail. So we are in the thylakoid within the chloroplast, and so inside the thylakoid is the thylakoid compartment. So that is the upper part of the picture here. Outside the thylakoid is the stroma, so that's the lower part of the picture, and it's divided. Those two are separated from one another by the thylakoid membrane. Embedded in the thylakoid membrane are the components that make up the photosynthetic apparatus. A little bit like the electron transport chain was embedded in the inner membrane of the mitochondrion. So here's how it happens. Light is absorbed by antenna molecules that absorb light at different wavelengths. That light excites electrons. As a result of the exciting electrons, we oxidize water and we create oxygen. Those electrons then flow down an electron transport chain, just like we've seen before. And it starts out in photosystem 2, or PS2. The full name's indicated here at the bottom. PS2, the photosystem 2, that's the apparatus, the protein apparatus that absorbs light. Between the photosystem 2 and the next big apparatus, which is photosystem 1, there are the elements of the electron transport chain. So the ETC flows here, and it is in the order of increasing reduction potentials, just like we've seen before. And the electron transport chain pumps protons. Photosystem 1 exists because once we get to the end of the electron transport chain, light is used one more time to excite electrons again. And the reason that is done is so those electrons then can flow downhill and actually reduce NADP plus to NADPH. So remember, NAD and NADH had a reduction potential of about minus 0.3. So what we're doing is we're exciting this electron up so that minus 0.3 is downhill, is a bigger electron reduction potential than is the electron here after it's been excited by photosystem 1. That allows the electrons to be captured by NADPH and to be available then for use in the dark reactions to fix CO2 into glucose. Okay, so excite electrons once in photosystem 2. Let them flow down the electron transport chain. Use that to pump protons. Excite the electrons again in photosystem 1 and let those flow without pumping protons, but simply to reduce NADP plus to NADPH. So those are the elements of the system. There are perfect parallels with the electron transport chain shown at the bottom of this slide. So you can compare the two. I'll show you at the end of this video, or actually in the next video, a direct comparison between the two. Kind of aligns the membranes and shows you how things work. Okay? But for now, take a look at this, compare the two. The light reactions are known as the Z scheme. So you can kind of see this if you look at the yellow arrows in this slide. You see that they form a Z in a sense. If you're looking sideways, there's the Z that I just traced out. So this is the Z scheme. Photosystem 1 is our first complex. Photosystem 2 is second. Between is the electron transport chain shown here. There's another electron transport chain we said after photosystem 1, but that does not pump protons. The electron transport chain between photosystems 2 and 1 pumps protons. So those are going to be pumped here in this part of the process. Okay, let's look at some detail in step 1 in photosystem 2. So light energy comes in, excites the molecules in photosystem 2. Now, it ultimately excites this P680. That refers to a pigment, P, that absorbs light at 680 nanometers. So 680 nanometers is the wavelength of light that is absorbed by this particular pigment. That pigment is key because that's the one that does the primary exciting of electrons up to an electron acceptor, and from there the electron transport chain is going to start. So before you get to P680, and that's a particular energy level that corresponds with the energy of 680 nanometers. Too bad my writing is not a little bit better. But the energy level of P680 corresponds to the energy of light with a wavelength of 680 nanometers. Now, photosystem 2 can absorb light of different energies, and those are all these green circles that are shown as absorbing light. And these are antenna molecules, we call them, and they absorb light of different wavelengths. Different antenna molecules absorb different wavelengths, and those excite electrons. As electrons are excited, they are passed to different pigment molecules. Typically, when an electron is excited to a higher energy state, when it falls back to the lower energy state, it releases some heat. And as it travels between these pigments, it is basically traveling between different energy states, pigment molecules that absorb energy, the light energy of different wavelengths. So it's a way of harnessing, of pulling together in an antenna a whole bunch of different wavelengths and then channeling those wavelengths through a series of pigments until the energy is finally delivered to P680. P680 is the key because that's the one that I said is the main electron exciter that starts the electron transport chain. But the pigment molecules are there to enable us to take advantage of energy of different energy levels. P680 can only absorb 680 nanometers. These other pigments might absorb 640 and 690 and 700 and 550 and whatever other pigments happen to be there. I don't know, I'm making those numbers up, but absorbs a lot of different wavelengths of light. So the electromagnetic spectrum, we've seen this, And we're talking about absorbing energy or absorbing light that's in the visible light spectrum. So if we absorb 680 nanometers, for example, we're up here in the spectrum, and we are absorbing red light at that point, maybe orange. Because we absorb red, what we see is the light that remains. And how that usually comes to our eye is as green. So that's why plants look primarily green. They absorb different wavelengths, but they absorb it in areas of the electromagnetic spectrum, which, when the rest of the light not absorbed is reflected, we see green. All right, step 2, we excite that electron from P680. So step 1, we absorb light and all the pigment molecules. Step 2, we excite that electron up to the primary electron acceptor. That is the step 2. So from there, electrons flow down the electron transport chain. Why do they flow? We've seen this before. The reduction potential is simply the tendency for a chemical species to be reduced. We have talked in class about how that is a characteristic of the particular chemical species, of the particular molecule. Some have more of a tendency to pick up electrons than others. We use the chocolate analogy in class. Some students like milk chocolate more than others. They have higher milk chocolate potentials in a sense. They're going to take milk chocolate from somebody who doesn't like milk chocolate as much, who has a lower milk chocolate potential. So reduction potentials are the tendency for a chemical species to be reduced. Reduction means gaining electrons. Oxidizing is losing electrons. This is a review. The more positive the potential, the more likely it will be reduced. So we measure that in volts. We've seen that before, and we'll see that in the next video when I show you the reduction potentials of the different electron centers in the electron transport chain in photosynthesis. In addition, we're going to talk about how much reduction potential it takes to pull an electron from water. That's pretty high. So we'll talk about how that works in the next video. Thanks for watching, and see you in the next one.